I am pleased to have Dr. Larry Wienkers here with us today. Larry received his Ph.D. in medicinal chemistry from the University of Washington in 1993 and completed his postdoctoral work in the Department of Drug Metabolism at the Upjohn Company before joining them as a research scientist. In 1998, Larry became Director of Drug Metabolism Enabling Technologies at Pharmacia and Upjohn, and in 2002 became Executive Director of Pharmacokinetics, Dynamics, and Metabolism at Pfizer. In 2004, Larry moved to Amgen, and he recently retired as Vice President and Global Head of the Department of Pharmacokinetics and Drug Metabolism. He is an AAPS Fellow with over 80 peer-reviewed manuscripts and book chapters. It is my pleasure to present to you Dr. Larry Wienkers.
Hey, thanks, Michael, for the introduction. And so it is kind of fun to be able to talk about some of the things that I've been observing lately with respect to drug metabolism. And so that's what we're going to talk about this morning. So when I joined the Upjohn Company back in 1993, our department was sort of known as Dr. Death, just like every other drug metabolism department. And for good reason. About 40%, at that time, about 40% of all the clinical candidates failed in development because of bad PK or drug metabolism issues, which kind of precipitated in the mid-90s kind of a renaissance where we looked to align our efforts very early in discovery, started doing a bunch of screening, and the consequence of that and understanding the various rates of failure, we're able to drive the rate of failure because of DMPK issues down to about what's right now hovering a little bit less than 5%. But you know, we're not kind of lulled into sleep, right? We're still a really, as far as the translational sciences group, drug metabolism is really one of the cornerstones of translational sciences. That is to understand everything associated with the drug, so that when we put new chemical entities into a human being, we've done our very best to underwrite the safety concerns. So this is sort of the outline for today. It's, compound developability and safety. Developability, I don't even know if it's a word. We use it as a word a lot, but that's essentially highly developable compounds, or compounds with low toxicity, no metabolic activation, high solubility, good membrane permeability, low protein binding, and predictable pharmacology. And then when we think about this is sort of our architecture. The first one and the fifth one sort of fit into this little flow scheme of the drug. Do we understand what its disposition? Do we understand its metabolism? Is it going to be reactive? Are the metabolites also going to be pharmacologically active? The middle three are really more physical chemical attributes that we can screen for very early. But essentially, in aggregate, what we're really talking about is DMPK scientists are constantly looking to pulling together a story of the drug and the consequences of metabolism. And let me show you what I mean. So if we took our poster child for metabolic activation, we would use acetaminophen, right? And if you were to just look at straight up acetaminophen, you'd say, how is this a drug, right? It gets bioactivated in NAPQI, and it binds to thiol groups across a variety of biologically important macromolecules. But because we tell the story of this drug, we know that most of it's eliminated through glucuronyl transferases and sulfonyl transferases, and that really the bioactivation of this molecule is driven by CYP2E1, but CYP2E1 is, ethanol induced, right? And we also understand that the glutathione, the thing that traps the NAPQI, is also variable with respect to poor nutrition and other issues. So I guess my point of this is, instead of halting the development of acetaminophen, we're able to context its issue associated with most of it getting metabolized through a non-toxic pathway. The toxic pathway is really attenuated by ethanol. So it's, you can read on the inside of it, it says if you drink, please don't take acetaminophen. And lastly, nutritional levels. So in aggregate, this is a very safe drug. People take it all the time because most of it goes through a metabolically stable pathway. The bioactivation can be minimized by not drinking, and we understand the risk elements with respect to glutathione levels to trap it. So it's a great drug, but there's a story behind why it's a great drug. Similar thing is seen with tamoxifen. Tamoxifen is a drug that's found great use, but we know that tamoxifen is metabolically activated into several metabolites. The major metabolite going to the end is methyltamoxifen, which is then subsequently metabolized by a variety of enzymes, including MOR3A4, but also, more importantly, 2D6, to lead to endoxifen. Endoxifen is really one of the primary drivers of activity, so it's a pharmacologically active metabolite, and you can see that the outcome, depending on whether you're a 2D6 poor metabolizer or an extensive metabolizer, you can see the Kaplan-Meier plot there for the outcome, and you pick up, an additional 20% outcome with this. And you can also see this on this path over here, where extensive metabolizers and intermediate metabolizers have a lot more of the endoxifen than the poor metabolizer, owing to what we're talking about here with respect to clinical outcome. Now, I bring up these two classic discussions because I really want to talk about here. We got this ICH-F9 guideline around metabolites. And basically what it kind of says is and I've heard this, now that I'm retired and going to a couple other companies, it's a little bit of a get out of jail free card. It's basically what I've been told is if we're developing our drug for life-threatening diseases, metabolism doesn't really matter. And so we kind of are doing a little bit of shortchanging our scholarship around metabolism. An example of this might be we sometimes fail to really understand the routes of elimination associated with major metabolites. Here's a classic example, kind of tragic really, because two molecules that looked like they had some really nice attributes for CMET turned out to be renal toxic because of the solubility issues associated with this metabolite right here. Well, we didn't pick this up because we didn't test the solubility of the metabolite because it probably wasn't synthesized. And moreover, the tox species, when we were looking to take this into GLP tox, was the dog. Well, unfortunately, as you guys already know, the dog doesn't have aldehyde oxidase, so they never produced this metabolite at any level in the tox species. And as a consequence, these two different molecules failed in phase 1 because of nephrotoxicity, and both were eliminated from further development. A similar story is here with respect to this molecule ozonosamid that we read about in the newspaper. Essentially, in 2015, Celgene bought this molecule for about $7.2 billion. It was going to be, best-in-class sort of molecule. What actually happened was one of the major metabolites that circulated almost at the same level, even higher levels than the parent drug, actually contained a significant amount of the activity, such that we didn't characterize the metabolite for activity. We didn't build a PK/PD model to understand the contribution of the metabolite. And so when they sent in the NDA, what they found out was they got to refuse the file letter, and of course you saw their stock dip. Now I've hence just checked it on yesterday. There's a PDUFA date for May 25th, 2020. But I'm just letting you see this, that because we didn't really understand what was going on with the metabolism and the activity of the metabolites, we had an untoward side effect of this was really disastrous to the company. So if we go back to home base again, We can add, there's one more additions that we should probably be talking about, and that is, in addition to low toxicity and pharmacological activity, we want to have low 3A4 liability. Now, DDIs are sort of passé, and I understand that, but they're still super important, right? They're still a major source. Like 1/4 of all adverse drug reactions are associated with DDIs. Most of the DDIs are associated with P450 inhibition. And so understanding the risk of the DDI is not only great for human safety, but it's also potentially a way to differentiate your molecule from other molecules in the same class. And you can see my little build there. You can see that even the product label running down the side, you know, the classic case is cimetidine. And we'll just take a sideline for this because I think it's always important to remind people of this. Cimetidine turned out to be the first billion-dollar-a-year drug in the United States. And it held its pole position for more than 5 years without a competitor on it. But it carried with it some clinically significant issues associated with drug-drug interactions with warfarin and many other drugs. So one of the molecule that came back, ridididine, was the second molecule in its class. And when that molecule came out, it was devoid of drug-drug interaction. So a couple of years onto the market, ridididine became the second $2 billion drug in the United States. And within 5 years on the market, ridididine became the first $2 billion a year drug, and cimetidine slipped into the $500 million sales class. And so I share this with you only from the standpoint of drug-drug interaction is one of the most facile ways to differentiate your molecule from your competitor molecule. So when we talk about drug-drug interactions, we immediately go to CYP3A4 as a victim, and for good reason. CYP3A4 accounts for about 50% of all the metabolism-based pathways associated with P450 oxidations, and it's probably the virtue of its massive active site. So you can see down here in this lower thing, these are the clinical interaction profiles, and you can see that, you know, CYP3A4 can bind molecules that are just a little bit over 100 Daltons, all the way up to 1,200 Daltons, like cyclosporin and such. So it's a very promiscuous active site. In the middle here, I kind of show you how it is a promiscuous active site, and you can see that if you were to think about the active site as a big room, and the heme as the floor, the ceiling of the CYP3A4 active site is covered with phenylalanine that kind of create this whole architecture, this dome above the active site. And there's very little amino acids protruding into the active site, sort of confounding or restricting access to the heme. How we know this is that we can do the phenyldiazine experiment with CYP3A4, where we coordinate phenyldiazine to the iron, reduce it, and that reduced intermediate can bind to the nitrogen, the pyrrole nitrogens of the protoporphyrin 9. And then down here you can see that when we do this experiment, reduce and have this thing hop off to have access to any one of the 4 nitrogens, you can see that we actually get almost a one-to-one relationship with all the nitrogens, suggesting that the active site is cavernous and there's nothing inhibiting or precluding access to the heme. So I bring the DDIs and the 3A4 as a context because this is where the game is right now, and everybody's very much into evolving cancer chemotherapy. We've seen a great uptick with respect to immuno-oncology and the success. And what we're starting to see now, and what the ambition is to sort of take cancer, and I say this in a very nominal way, but begin thinking about treating cancer in the way we treat a bacterial infection. You know, when we have a bacterial infection, it's because our T cells weren't able to keep up with the foreign object, in this case, the bacteria. And so the bacteria starts to win. So what do? We add antibiotics to sort of debulk the tumor, and we allow our T cells, our immune response, to take care of the rest of the bacteria, and that's how we live through a bacterial infection. In a similar case, we start to talk about immune responses and T cells by killing cancer cells, but combining that with debulking agents, targeted debulking agents, where we can have the 1-2 punch and really drive the therapeutic efficacy of chemotherapy. So one of the major pivot points in this whole strategy has to do with bringing in various tyrosine kinase inhibitors into the mix as our sort of targeted therapies. And you can see that for kinase inhibitors, in the last few years, there's been just a tremendous amount of effort and success in bringing all these kinase inhibitors to the market to give clinical benefit to cancer patients. Now, when we look at the kinase inhibitors, and this is why they're so tricky to work with, their active site and the nomenclature associated with it, and essentially the QSAR around these kinase inhibitors, all have a central feature. And that is, we have this hinge region where we have this nitrogen groups hanging off. We have something that's going to be great for solvent binding to water. And then we have a big piece of grease that hangs over here. And almost all of the molecules sort of fit this structure-activity relationship to one way or another. So keep this feature in mind here. So we've got water-soluble, we've got grease, we've got some essentially basic to neutral molecules that are, in sense, if you're thinking grease and water-soluble, that's an amphoteric molecule. So we have a neutral to basic amphoteric molecule. Well, that is exactly in the wheelhouse of CYP3A4, where the best 3A4 inhibitors in this case are high log P, electronegative, polarized at one end, amphoteric molecules. And you can see that when we looked at QSAR around these things, it's all, essentially, you couldn't have written a better diagram of a CYP3A4 inhibitor when you overlaid it upon the kinase structure. And so for the bulk, and I'm picking on these molecules, but almost every kinase, if you look in their section 7 of their DDIs, you can see that almost all of them are contraindicated with CYP3A4 substrates, that you have to have. There's potent inhibitors, and you have to have dose adjustments associated with all these molecules. And again, we could have 3 more slides of these molecules with this type of writing. So it's a real big deal. Part of the reason why it's a big deal is these amphoteric molecules, and many of you already realize this, are really susceptible to nonspecific binding in microsomes. So in this case, what we have are these molecules coming in. Because of high specific binding, they don't appear to have KIs and KMs. Their KIs and KMs are relatively high when you calculate it, unless you correct for nonspecific binding. And in many cases, this causes a 2 order of magnitude left shift. So what you thought was a 10 micromolar KI for your drug against 3A4 really turns out to be 100 nanomolar KI, and given the concentrations that you're going to be dosing, it's a big deal. Now, we've seen this for other types of molecules, in this case, ketoconazole. You can see almost a linear relationship between the milligram of protein associated in the incubation and the concordant KI associated with the measurement, all because of nonspecific protein binding in this case. And Scott Albach has also showed this, where depending on the amount of protein, you can have a left shift of the KI, of the IC50 associated with fluvoxamine, another molecule that's amphoteric, has got some grease associated with it. So a lot of companies have been completely surprised going into the clinic where they thought they had a clean molecule, and it turns out they have a potent P450 inhibitor, which was actually inconsistent with their target product profile. Another part of these molecules, these kinase inhibitors that are important, has to do with mechanism-based inhibitors associated with them. So I've got a little kinetic scheme up here to can remind you of, essentially the inhibitor binds the enzyme back and forth. There's an activation complex, and it either gets the inhibitor either gets oxidized and kicked out as a product in this case, or it binds irreversibly and essentially destroys the P450 by covalently binding the apoprotein or knocking out the heme activity, but essentially renders the enzyme inactive. It's a little bit old now, but you can see a bunch of the molecules that are known to be on the market and are known to be 3A4 or known to be P450 mechanism-based inhibitors. And I've highlighted in yellow all the ones that are associated with 3A4, and it kind of makes sense, right? 3A4 owns the bulk of drug interaction, so why would it also own the bulk of mechanism-based inactivation? And so we can talk about this, and what we have is there's three mechanisms of P450 inactivation. One is through heme destruction, where you get bound, reactive intermediate binds to the heme and destroys, in this case from a diagnostic experiment, the CO binding spectra. You can get apoprotein induction, where the molecule gets activated to an electrophile, binds to some amino acid within the active site, and what you can see here by mass spectrometry is a mass increase associated with the enzyme plus the adducted protein. And lastly, the molecule can complex with the heme iron. In this case, this is called pseudo-irreversible, but for all intents and purposes on our time scale, it is irreversible. And this is where we have the diagnostic MI complex, as they call it, or a 455 spectrum. So I pulled together as many as I could fit where you can still see the structure, kinase inhibitors that are known to be mechanism-based inhibitors as well. Again, you can see the classic attributes of all these kinase inhibitors. So they're all bound, looking for metabolic activation. And so let's dive into what might be going on. Well, one of the features of these molecules that you've already seen is that in addition to CYP3A4, aldehyde oxidase also has the same type of QSAR associated with binding to these molecules. And you can see that there's a bunch of phenylalanine rings and neutral molecules within the active sites. We're starting to see pi-pi interactions. And unlike P450s, and if we go over here, we can see that the P450s are really kind of lazy enzymes. They take the easiest approach for the oxidation. And so as you can see, most P450 reactions have to do with ND alkylations. And then as it energetically gets more disfavorable, these are sort of become secondary metabolites with respect to, pulling off hydrogen off of a central carbon here. But you can also see that the overlay between CYPs and aldehyde oxidases are massively similar. The metabolites are dissimilar, but the reactions and the ability to bind the same types of molecules are incredibly similar. So why is that a big deal? Well, in this case, you can see that these molecules have shown that we had reasonable KIs. But when we went into the clinic, we found that we had huge changes in victim drug AUC. And what happened here is that we accounted for the KI associated with the molecule, but we didn't account for any metabolites that could be bioactivated or actually serve as inhibitors as well. So we did our PBPK model, we plugged in our KI, and we said we should probably be relatively clean. And in the clinic, and all of a sudden we see a 5-fold increase in midazolam. And a similar case here where we had reasonable KIs, but the KNX drove almost a 20-fold increase in activity. So point being is a facile run through of just the characterization of the Ki, plugging the Ki, whether it's accounted for unbound or not, into the PBPK model without taking into account what the metabolites look like and understanding major metabolites contributing causes some pretty big surprises early in the clinic. So what might be going on here? And so one of the things that very seminal, the CYP3A4 mechanism-based inhibitors, as molecules that might be a Michael acceptor. And so let's just do the chemistry of a Michael acceptor here. You know, what we're talking about is just creating a, sort of an acrolein species here, where all of a sudden there's a reactive position and in a thiol group, and you can have glutathione or any other cysteine within a macromolecule binding to this in essentially an irreversible fashion. And so you could see, for an example here, we see a defluorination by a P450 of sunitinib having this, but this is not really the metabolite. It might be the metabolite you see in the mass spectrometer, but if you close your mind's eye, all of a sudden you can push these electrons through to form this reactive intermediate, and this is the activated species that's associated with mechanism-based inactivation of sunitinib. Probably trying to drive this down and maybe a little bit too much, but I think it's worthy to think about. So here's AMG 487, which was shown to be a 3A4 mechanism-based inhibitor. One of its major routes of metabolism was M2, where we had an O deethylation to form this species. But as you can imagine, this species is one electron away from forming this species here, right? This Michael acceptor. And so what we've seen is covalent modification of CYP3A4 of this species to cysteine 239. Cysteine 239 is right in the egress channel of the 3A4 active site. So 3A4 is pretty interesting with respect to that, the reactive thiols, because what we can see here is there's actually 7 cysteine groups in CYP3A4. One, as for almost for all the P450s, mammalian P450s, coordinated to the 5th axial ligand of the heme, sort of the anchor of the heme. But cysteine 239, 468, and 98 have all been shown to be susceptible to bioactivation and covalent adduction. And here's just another example of that, where we had raloxifene getting bioactivated to a reactive species, binding the cysteine 239 within the active site. And you can see as you go through the egress channel. So this is sort of a hook within inside the active site of a metabolically activated species that it can bind to. So I bring this up only from the standpoint of, we have CYP3A4, it's a large active site. We might not see the activated species coming out of CYP3A4, but because aldehyde oxidase really doesn't change the lipophilicity and the character of the molecule, a metabolite stemming from aldehyde oxidase can also very easily slip into the CYP3A4 active site, where it's only one oxidative step away from becoming a reactive intermediate. And this is seen here with idezolib, where you can see it's a great CYP3A4 molecule inhibitor, but it's really all driven by its K-inact, and this is the species that's known for it. So if you have this species, again, one oxidative step away here to over here to forming an electrophilic species. Another molecule, we can go through the same pieces. Metabolic, here's your spot where it's going to be aldehyde oxidase creating this species. Again, imagine just dropping an electron into this species to create a Michael acceptor. These folks did a very thorough way to understand the metabolic activation of this. So besides doing the KI very selectively in microsomes, accounting for free fraction, They also went into hepatocytes, which is a very strong way to understand metabolism-based inactivation. So you can see here they have the time dependence, or the IC50 shift in hepatocytes. However, when they incubated hydroxylene with the hepatocytes, and remember hydroxylene is an inhibitor of aldehyde oxidase, you can see that they essentially evaporated the IC50 shift associated with this. These guys, hats off to them. They must have had a great working relationship with their medicinal chemists, because they were actually able to synthesize deuterated VX509 and show that the deuterated species also block the metabolic activation and mechanism-based inactivation associated with CYP3A4 destruction. And as you can see, basically the deuterium isotope effect for aldehyde oxidase for that type of reaction is about 5. So, again, just showing that the molecule itself in microsomes might not have led you down a path to think it's going to be a mechanism-based inhibitor. Running it in hepatocytes and seeing it is the first sort of red flag associated with the molecule. But then if you start to build upon the story, you begin to walk down the pathway of, okay, it's aldehyde oxidase. How much is that going through this particular pathway? You begin to mitigate the risk by building the story of the molecule and not running through a sort of a checkbox activity associated with the metabolism. So it's kind of a big deal as drug metabolism scientists that we build and continue to build the story of the molecule, especially as we start to come out of discovery and we start to call it as a clinical development candidate. And because you don't want to have this stuff happen on your watch, right? And just looking at the molecule, you're not going to be able to pick out where the metabolic activation is occurring. And my favorite story about saying that has to do with furaflin. Back in the mid-90s, furaflin was found to be a mechanism-based inhibitor of CYP1E2. Looking at the molecule, everyone in the literature was convinced that this furan ring was the responsible species for inactivated CYP1A2. However, when detailed kinetic experiments were carried out on furaflin, Kunze and others were able to find that it had nothing to do with the furan ring. It had to do with the metabolic activation of this species, dropping in and creating essentially an electrophilic species that then covalently bound to the nucleophile within the active site. So again, we can't armchair metabolic activation, but if we begin to build our story, trying to investigate the molecules, if it's early enough, we can feed back that information to the medicinal chemist, and we can sort of steer our way away from these problems. So getting back to what we were talking about, understanding the free fraction or the unbound fraction in the microsomal incubation and setting that into context with your calculated Ki is incredibly important to predict, and that's the number that needs to be plugged into the PBPK model if you're going to predict just how big of an inhibitor it's going to be in vivo. So a cavalier approach to understanding KIs for these types of molecules means you're going to be wrong. So understanding the story and doing the next experiment to really understand the molecule will sort of keep you out of harm's way professionally, because if the big inhibition crops up, but you had already given the molecule a clean bill of health, the organization kind of looks back at you, especially when they are kind of encountering these clinical surprises. So, you didn't put in your target product, your product target profile that we wanted to have a potent P450 inhibitor. So getting these things done early, understanding it with the appropriate high fidelity information will allow you to develop the right type of molecule. And lastly, if you begin to understand the chemistry and the metabolism that's going on, you may be able to fit, working with the chemist, different mechanisms to block bioactivation. So, here we are with this species, K3 delta kinase inhibitor that had a potent TDI. However, what they were able to do was hang essentially a blocking group on this site where AO was going to be activating it, and they were able to completely abolish the mechanism-based inactivation associated with these types of molecules. So again, understanding the pathways, building the story around the molecule, and then working with medicinal chemists, you can completely avoid these liabilities with these types of molecules.
All right, thank you, Larry. While we wait for questions to come in, I wanted to mention that information about our services and test systems, published papers, posters, other webinars, and more is available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding Larry's presentation or any of Saxui XenoTech's contract research services or products following the Q&A, please feel free to ask them in the post-webinar survey or contact us through our website. First question that came in was, what is the typical success rate for identifying all the metabolites of a new drug? How often does the drug get converted into something that cannot be picked up in the metabolite screen? What is regulators' requirement for identifying all metabolites?
That's a great question. So, we typically are for looking at metabolism, our go-to molecule, our go-to system is always microsomes because of cost and just ease of use. But what I'd say is, you look for a metabolite in the microsomes, but then you don't stop at microsomes, and you go ahead and for especially specific molecules, make sure that hepatocytes, it's sort of like making sure that the cuff, if you were, making sure that the cuff matches the collar. And that's sort of, a seamstress thing. But basically, if you're running hepatocytes and you see a completely different metabolic profile than microsomes, then microsomes are only giving you half of the story, right? And so you need to drill down into, and so, you may have to use hepatocytes as your screening tool, or at least a secondary screening tool to ensure that you're capturing the metabolites. Now, with respect to the regulators, obviously metabolites and the major metabolites as it's going forward, as you're heading toward GLP studies, you're going to want to make sure that the monkey hepatocytes, the rat hepatocytes, the human hepatocytes, and the dog hepatocytes all line up. And then from that, you can choose your tox species. Now, if you, like I said, if you had if your molecule was P450, but also aldehyde oxidase, and aldehyde oxidase was contributing significantly, or at least maybe not even significantly, meaningfully to the disposition of your molecule, dog is a pretty bad tox species, right? Because the dog's not going to produce that metabolite, so that coverage isn't there. So, getting concordance between the microsomes and the hepatocytes is super important. If everything looks the same, you can always be fooled with extra hepatic metabolism and such. But right at that gate, you can stick with microsomes and feel pretty confident. But if the microsomes don't reflect what's going on in the hepatocyte, you've got to rethink your strategy and begin to find ways of incorporating the hepatocyte data, the total metabolism package associated with the drug, before you head off into, important studies like GLP tox studies and such. You wouldn't want to go into the clinic having a discordance between microsomes and hepatocytes, because while you could get away with it being exactly right, there's a high probability that you're going to be wrong and there's going to be a surprise. And I think we owe it to patients to make sure that we're not really generating surprises. Hope that helps.
All right, a couple of other really early on questions here. What are major FDA concerns on metabolites for an end of phase 2 meeting?
Well, I think that the end of phase 2 meeting is, you've already sort of passed the safety. You've driven concentrations up in phase 1 plenty high. You're not going to see those concentrations again. I think that the metabolites, are any of the metabolites active? Part of phase 2, in my mind, is you're going to head into phase 3, and so you're going to be proposing the clinical dose. And so if you have metabolites that are circulating that have activity, have you incorporated into your PK/PD model the contribution of the parent? Of course you have. But is there any contribution from the metabolites? And why I bring it up that way is the P450s have a lot of interpatient variability, which impacts the clearance of the molecule and exposure to the parent molecule, of course, but it also has a lot to do with active metabolites. So understanding the activity and whether any of the metabolites are contributing to the overall pharmacology observed in the clinic is, one of the places where you want to make sure you batten down the hatches and you can speak meaningfully to that. So, is there, for major metabolites, and you don't have to do this in a GLP setting, you can do this, in a GLP assay, you can do this in non-GLP. Are there any metabolites circulating, whether at the same concentration of parent or lower? I'm not talking only just about disproportionate metabolites. You could have coverage in the tox species, but that still means you might have to understand the presence of the metabolite as it contributes to pharmacological activity. I think there's two swim lanes that are going on with metabolites. One is the misguidance. Have you accounted for it in the tox species? Does it represent X amount of a particular of the parent drug or total drug related species? So yeah, that's one swim lane that needs to be accounted for. That's pretty straightforward. The other one, that if you've passed that and all of a sudden you can't turn a blind eye, you could have metabolites that are circulating, and if they have activity, they can be contributing to the pharmacological benefit of the molecule. Now that's a big deal, that if you were to be able to add that into your PK/PD model, that would give the regulatory agency great comfort that you understand essentially that the appropriate dose, the interpatient variability, and what's contributing to pharmacological activity. So you can pass the MIST, but still want to understand the presence and the pharmacological activity associated with some of the metabolites.
Does AO involvement in metabolism lead to primate as choice as second species in tox?
I would say a lot of times yes, but you can context that with respect to how much is going through AO. The one thing I would say is that aldehyde oxidase, P450s are a pretty inefficient protein. Their rate of oxidation is 1 a minute, right? It's not super high. Their kcat is not super high for P450. It is really high for aldehyde oxidase. So if a molecule is a substrate for aldehyde oxidase, a lot of times it's going to be a really good substrate and it's going to get turned over fairly well. So yeah, I mean, the second species as if it's AO and it's a meaningful pathway for AO, yeah, going cyno is the right way to go, because again, you're appropriately de-risking the molecule before you put it into people.
Are S9 fractions a reasonable way to explore the activity of AO in place of hepatocytes?
You can get away with it, but this is the case where you have to do the right experiment. And I've seen, talk to a bunch of people. They bought their S9. They didn't see an AO metabolite, and you go, "Gosh, that sure looks like an AO substrate." And then you ask that question to everybody, "Hey, what was your positive control? And they didn't have one. So lack of activity doesn't mean, lack of AO metabolite doesn't mean no AO activity unless you were able to run a positive control and show that you had a strong AO activity within the S9. So yeah, I think you can do S9. You know, I think it's fine, but make sure that you've got the right controls as you're doing it if you're going to characterize it. Not all S9 has great AO activity.
For a potent 3A4 MBI with a short T1-2, which is dosed intermittently, say once a week, how long would 3A4 be inactivated P450, i.e. could you dose 3A4 substrates a day after the MBI was dosed?
You know, you might not. And I'll say this is that, you know, when we talk about kinetics, we have, you know, if we talk about reversible kinetics, we're either impacting the substrate's ability to get into the active site with a competitive inhibitor, and that's an impact on Km. Or we talk about a non-competitive inhibitor, which binds to the protein some other place, and it makes the protein less efficient, which is a K-cat. But when we talk about mechanism-based inhibitors, we're taking the E total. So remember, if you remember back to Biochemistry 101, Vmax is essentially total amount of protein times K-cat. So again, if we go back to the non-competitive, we're changing K-cat, but we're not changing protein, such that when the non-competitive inhibitor goes away, the enzyme's back up to 100%. With a mechanism-based inhibitor, we're impacting E total, and total amount of enzyme. We can take that to 0. So now you have to wait for the synthesis of, let's say 3A4 to come back. Now the synthesis, the half-life is somewhere between, depending on the literature, and I hate to say it this way, but it's between 26 and 80 hours is the half-life. So, you know, if we're talking about knocking out all the CYP3A4, it might be several days. If we're taking the shortest half-life, the synthesis rate possible, it might take 4 days to get back up to 100%. So in that case, a day after is going to, you're going to have a significantly less 3A4 than you had in controls. I hope that helps. But you know, if you look back at the synthesis rate, and again, I say it in terms of just thinking about it very simply in kinetic terms. Michaelis-Menten kinetics is always about having the same amount of protein throughout the incubation, and that all works. Mechanism-based inhibitors take out the protein. And so that's why PBPK models with reversible inhibitors are really great. PBPK models associated with mechanism-based inhibitors are less predictive, meaning the outcome doesn't really match as well as we'd like it to do. And that's because we're really talking about resynthesizing protein and getting E total back up to what the control was. And that might be different for different P450s, and it might be very different for different individuals based upon their age and other factors. So I hope that helps.
In terms of PPB, what is acceptable? Some known drugs are greater than 99% PP bound. Is the relationship between on-target potency, IC50, on the intended target, and PPB meaningful? Or plasma protein bound drug can act as a depot of the drug in the circulation?
Yeah, I mean, a lot of the protein plasma binding of the drug is going to be the alpha-glycoprotein and albumin. And, you know, there's specific regions in those that have high affinity, and that's where a lot of, you know, like if you look at warfarin, that's 99.99% protein bound, it's bound up really tight to a binding region within albumin. It doesn't reflect the non-specific protein binding associated with the incubation. Again, what I'm talking about is those amphoteric molecules that are neutral and basic that just like to go into the grease, the chopped up endoplasmic reticulum that essentially the lipid that the P450, the reductase, and the B5 swim in, these particles, that these molecules can just get caught up in something that's, again, non-specific binding. It's just getting caught up in the grease. And so you can have high plasma protein binding and have low non-specific binding in microsomes, but you can also have high plasma protein binding and high non-specific binding. So it's really examining the molecule, and one doesn't lead to the other. One, in many cases, a very elegant interaction between the molecule and albumin binding site and versus a very vulgar non-specific binding that has to do with just the chemical structure and the associated, essentially physical chemistry of the molecule.
All right, I know we're pretty much out of time, but maybe we'll squeeze in a couple more questions, and then we'll have to take the rest of them offline. To help detect complex MDI, do you advocate measuring CYP activities as part of a CYP induction study in human hepatocytes?
I think trying to get two endpoints out of one study is oftentimes fraught with error. I think that if you had a well-controlled studies and you're able to do it, I think that that's an okay way. But sometimes making experiments more complicated lends itself to more hand-waving.
In general, do you recommend TDI screening for multiple P450 isoforms or 3A4 plus specific ones on a case-by-case scenario?
I think the latter. I mean, you're gonna run your TDI and microsomes, and that's going to tell you if there's anything going, with your specific inhibitors. Essentially you're doing your IC50 shift across a bunch of different P450s. If that's there, then you might be dropping it into hepatocytes as the molecule starts to evolve. Essentially, and this is the way I always think about it, at least, is you can think of a project as a train sitting at the station. There's a whole bunch of stuff that you can do before the train leaves. No one if the train leaves the station, it's moving. And all of a sudden, doing those experiments and having those experiments impact the development of the molecule become less meaningful because the train is leaving the station. So if you're going to get after it, you have to get after it awfully early. So if you're a clean DDI or TDI with the microsome, dropping it as just a de-risking into the hepatocytes and making sure that the story holds is the great way to go. And so I think that you'd feel awfully DDI or TDI associated with your IC50 shift, a safeguard before the train starts to leave the station, so to speak, and hepatocyte is probably meaningful, and it's actually relatively cheap activity. And even if you see something, your organization may choose to stay the course with the molecule because you could put it into the appropriate context. But moving forward, essentially everything that we do in drug metabolism is part of a translational sciences focus is to build clinical conviction associated with the molecule. Do we feel good about putting this molecule into human beings? And so the little things that we can do to build the story, to provide context around the molecule is exactly our job.
Okay, this wraps up today's session. Thanks to all of you for taking time out of your day to join us, and thank you to Larry for that great presentation. Have a great rest of your day.